a lot of different ways have come forward that are interesting in terms of looking at clonal diversity or intratumoral heterogeneity. And many of these have been bulk next generation sequencing assessments at various time points in patients' therapies. But what you haven't been able to look at when you use that technology is what is the actual makeup of individual cells within these clones, and how does that change on a cell-by-cell basis, rather than a snapshot of the heterogeneity of the marrow for mutations. What are the actual mutations in individual cells? And the reasons this might be important is there could be cells that have a mutant copy of a gene but no wild type, or cells that have no mutation, just wild type, and if you average those two together, it looks as if you had one cell that has just mutation, one cell that has just wild type. It looks like it's a 50-50 split. You don't realize there's actually diversity there. And so with the mutational complexity of AML, you don't actually get a sense of what is truly in the marrow. Why does that matter? It matters if you're trying to track for clones across therapy to see how these rise and fall and what the heterogeneity of the marrow is. And if you're looking at targeted agents, you may be able to spot resistant populations very early in therapy and either watch these expand or contract in terms of a clone responding to a targeted agent. So there's many ways that we want to look at this. Recently, the technical ability to look on a cell-by-cell basis was developed, and the presentation is by Dennis Eastburn, who's worked with a platform called Tapestry. And Tapestry basically is a way to encapsulate cells for an instrument that's able to then barcode the DNA after protease digestion of the cells, and that basically allows you to identify what DNA came from what cell, because it all happened within the reaction in an encapsulated cell at that time. A next-generation library is created, and then you can run next-generation sequencing, get a panel of readout, and then know from the barcoding where those came from. So literally you have a next-generation panel on a cell-by-cell basis. The instrument runs about 10,000 cells at a time, and you can look at that for very small clones and be really quite certain that what you're seeing is actually what was there. So we looked at this in terms of following this for patients that were treated across therapies, including targeted therapies, including Gilteritinib, again, which was recently approved for therapy of patients who have FLT3 mutations who've relapsed. And we can watch during that therapy what happens to the clone size. As we're seeing clinical response, are the clones collapsing, expanding? Are there resistant populations that are growing? And we're learning a lot about what actually happens under the selective pressures of a FLT3 inhibitor that's quite selective for its target. So this has been an interesting way that we've looked at our patients during therapy, not so much to pick one therapy over another, but to better understand how intratumoral heterogeneity changes in the context of a targeted inhibitor. And we hope to have data for this published soon.